Can I get confirmation from someone that my screen is shared? Yes, yep. Wonderful, thank you. Thank you so much. Okay, so hi, my name is Rachel Rigden. I am a PhD student in Dr. Mark Cadot's lab at the University of Toronto in the Department of Physical and Environmental Sciences. Today I'm going to be giving a catalyst talk about my ongoing analysis that I'm conducting using intraspecific trait variation to predict a species persistence over a gradient of invasion here in Toronto at Rouge National Urban Park. Before I begin, I would like to acknowledge that this work would not have been possible without two Cadot Lab alumni, Dr. Stuart Livingston and Darwin Sodhi, who collected the data throughout the analysis in 2016. So understanding what factors determine a native species sensitivity to species invasion is critical to making predictions on how invasive species will affect a community or an ecosystem. And this question got me thinking about the role of functional diversity and trait variation, specifically within species trait variation in determining invasion success. Functional diversity is defined as the variation of traits in a functional space and is a component of biodiversity, highlighting the role or function a species plays in the ecosystem rather than just what species it is. Intraspecific trait variation is the trait variation at the individual level. For example, there's about 60 people in here right now, and we are all not the same height despite all being humans. We vary in our heights. Intraspecific trait variation contributes greatly to functional diversity, and even though it's overlooked, it's often overlooked, it plays a large role in community and ecosystem functioning. Here, we tested the role of intraspecific functional richness and intraspecific functional dissimilarity to an invader to predict a species' sensitivity or persistence over a gradient of invasion. Our study invasive species is Vincetoxicum rossicum. As you guys saw in the last presentation, it is a very concerning invasive species in the Greater Toronto area. It is a perennial herb native to Ukraine. It is invasive in parts of Ontario, Quebec, and northeastern United States. Vincetoxicum rossicum has detrimental effects on native plant species and has the potential to displace native species and dominate the landscape. We tested our questions at Rouge National Urban Park in Toronto, Ontario. Rouge Park is one of the world's largest urban parks. We have 13 open meadow sites dispersed across the park, ranging over a gradient of Vincetoxicum rossicum invasion. Relative plant cover, so percent plant cover, and trait data was gathered for each site in 2016. Some of the trait data was collected for 36 abundant species. Some of these species are native and some are non-native, and this was taken into consideration throughout our analyses. We gathered trait data for 5 traits: specific leaf area, leaf dry matter content, plant height, leaf carbon content, and leaf nitrogen content. Each of these traits is correlated to a functional link. So specific leaf area is correlated with relative growth rate, photosynthetic rate, and leaf nitrogen content. Leaf dry matter content is correlated to relative growth rate and leaf lifespan. Plant height is correlated to reproductive size, fecundity, potential lifespan, and resilience. Leaf carbon content is correlated to carbon inputs in the soil, herbivory rates, and biomass production. And lastly, leaf nitrogen content is correlated to photosynthetic rates and relative growth rates. Using trait probability densities and in the R package TPD, which is described in Carmona et al. 2019, we calculated intraspecific functional richness for each trait among the 36 different species. Intraspecific functional richness is defined by the amount of functional space occupied by a species. In other words, it is the breadth of variation for each trait for each species. In addition, we used trait probability densities to calculate the species dissimilarity to Vincetoxicum rossicum for each trait. So how are they similar to this invader, Vincetoxicum rossicum? And to do that, we subtracted the overlap of functional space or richness from 1 for each species compared to the invader. We compared these intraspecific functional metrics to species persistence over an abundance gradient of invasive Vincetoxicum rossicum. To estimate species persistence to Vincetoxicum rossicum, we conducted a series of linear regressions. So we compared relative Vincetoxicum rossicum abundance to relative abundance of each species. And that slope that we found, we made that signify species persistence. We found that intraspecific functional richness of leaf percent carbon and leaf percent nitrogen predicted species persistence over an abundance gradient of Vincetoxicum rossicum. And looking more closely, there was a negative relationship. So first, we can look at leaf nitrogen content and see that intraspecific functional richness, as intraspecific functional richness increased, we saw a decrease in species persistence. And furthermore, if you separate the species into exotic and native species by origin, you see that native species are driving this response. And similarly, with leaf carbon content, we see this negative relationship between intraspecific functional richness of leaf carbon content and species persistence. And once again, we see that native species are driving this response. In addition, we found that an increased intraspecific functional dissimilarity to Vincetoxicum rossicum was for just leaf percent nitrogen was associated with an increased species persistence over an abundance gradient of Vincetoxicum rossicum. You see this positive relationship between leaf nitrogen content and species persistence. So as dissimilarity to Vincetoxicum rossicum increased, we saw an increase in persistence. And that means that the more that the leaf nitrogen content differed from the invader, the more likely the species was to persist over an abundance gradient. So in summary, we found that increased functional richness was associated with decreased species persistence over an abundance gradient, particularly for leaf percent carbon and leaf percent nitrogen. In addition, we found that intraspecific functional dissimilarity to Vincetoxicum rossicum was associated with an increased species persistence over an abundance gradient of invasion for leaf percent nitrogen. These results suggest that intraspecific trait variation does play an important role in species invasion and plant invasion success, and highlights the importance of considering in the future intraspecific trait variation when assessing the impact of species invasion. And with that, I'd just like to thank you all for listening to me today, and any input is appreciated.